Hello guys, welcome to another video. In this video we are going to learn about the restriction enzyme. Restriction enzymes are the enzymes produced by certain bacteria that have the property of cleaving DNA molecule at or near specific base sequences. Here you can see in the picture specific base sequences, restriction enzyme cleave the DNA. So this is a restriction enzyme. So here are some properties of restriction enzyme, such as a protein produced by bacteria that cleaves the DNA at specific sites known as the restriction site. The restriction enzymes protect the live bacteria from bacteriophages. When the restriction enzyme recognizes a DNA sequence, it hydrolyzes the bond between adjacent nucleotide and cuts through the DNA molecule. So these are the properties of restriction enzymes. Then derivation of the EcoRI name. What is the meaning of EcoRI? E means Escherichia, which means the genus. Then co means coli. Coli means specific species, and R means RY13, which is known as strain. Strain is RY13. Then I refers to first identified number, orders of identification in the bacterium. So this is the meaning of EcoRI. That is genus, species, strain, and order identification. EcoRI. Then types of restriction enzymes. There are mainly 6 types of restriction enzymes. First one is type 1, type 2, type 3, type 4, type 5, and final is artificial restriction enzymes. So let's go to the type 1. Type 1 restriction enzymes cut the DNA far from the recognition sequences. Very common in genomic analysis, used mostly for biochemical interest. Then next one is type 2. These enzymes cut at specific position closer to or within the restriction sites. They are exclusively used for DNA analysis and gene cloning in the laboratories. Then type 3. Type 3, these are multifunctional proteins. There are 2 subunits such as RAS and MOD. It is modification methyl transferase. Then type 4. Type 4 restriction enzymes only cleave the methylated and DNA and will not act upon the methylated DNA. ERO-KM CRBC, the only type 4 restriction enzyme commercially available. That means this is the only commercial available type 4 restriction enzyme. Require 2 hub sites for successful DNA recognition. These sites can be separated by up to 3 kilobase pair. Then here we can see the compare among the type 1, type 2, type 3, type 4. Here you can see the factors such as structure, recognition site or pattern, cleavage site, cofactor. Here are such as type 1 structure is heterotrimer, type 2 homodimer, type 3 homodimer. Then recognition site, type 1 2 sites, type 2 small, type 3 2 sides, head to head, orientation non-palindromic, and type 2 is palindromic. Here we can see different types of comparison among the type 1, type 2, type 3 and type 4. And here are some cofactors such as type 1 for this, type 2 for this, type 3 for this, and type 4 for this. And type 5 utilize guide RNAs to target specific non-palindromic sequences. They can cut DNA of variable length, flexible, and ease of use. And the last one is artificial restriction enzymes. It is generated by fusing a natural or engineered DNA binding domain to a nucleus domain. Artificial ribonucleases that act as restriction enzymes for RNA developed by PNA based system which is similar to DNA or RNA. So this is about the artificial restriction enzyme. Depending on the cut, restriction enzymes is two types: blunt end cuts and sticky ends cut. Blunt ends. Some restriction enzymes cut DNA at opposite base. They leave blunt-ended DNA fragments. These blunt-ended fragments can be joined to any other DNA fragments with blunt ends. Enzymes useful for certain types of DNA cloning experiments. Here you can see the blunt end cuts. Then sticky ends cut. Most restriction enzymes make staggered cuts. Staggered cuts produce single-stranded sticky ends. DNA fragments with complementary sticky ends can be combined to create new molecules, which allows the creation and manipulation of DNA sequences from different sources. This is sticky ends cuts, and that is blunt end cuts. Then palindrome and non-palindrome sequence. Palindrome means the mirror-like palindrome in which the same forward and backwards are on a single strand of DNA strand, as in GTAATG. The inverted repeat palindromes is also sequence that reads the same forward and backwards, but the forward and backward sequences are found in complementary DNA strands, GTATAC being complementary of to CATATG. Inverted repeated palindromes are more common and have greater biological importance than mirror-like palindromes. That means palindrome means mirror-like structure, and here you can see that GATC and mirror-like structure GATC. This is mirror-like structure, and the opposite of palindrome is non-palindrome. That means non-palindrome is non-mirror-like structure. Non-palindrome means opposite of the palindrome. Then here we can see some enzymes, their source, their recognition sequence, and the cut. First enzyme is EcoRI. EcoRI means Escherichia coli, and the recognition sequence is 5' to 3', and the cut is 5' to 3'. Then TAC-on means Thermus aquaticus, and the recognition sequence is this, and cut is this. Then PVU2, that is Proteus vulgaris, and the recognition sequence is this, and cut is this. Then small, that is Serratia marcescens, and the recognition sequence is this, and cut is this. And then EcoRI is Escherichia coli, and the recognition sequence is this, and cut is this. And then last one is XBA-on, that is Xanthomonas badri, and the recognition sequence is this, and cut is this. So this is the list of some enzymes, restriction enzymes, their source, their recognition sequence, and their cut. Then last slide is application of restriction enzymes. There are some applications such as recombinant DNA technology. They are used in RFLP techniques, that is gene mapping, construction of genomic library or cDNA, then sequencing DNA used for gene cloning and protein expression experiments. So these are the application of restriction enzymes. So that's all about the restriction enzymes. that's all for today. See you in the next video. Thanks for watching.